This video provides an overview of the visualization of the origins of DNA replication. Replication in eukaryotes involves many origins of replication and is active during the S phase of the cell cycle, which lasts approximately 8 hours. This raises two important questions. Are all origins of replication activated at the same time? And is there any coordination between the replication timing and the type of chromatin? To answer these questions, analysis must be conducted on synchronized cells, and the replication sites need to be visualized. Cell synchronization is crucial for meaningful observation, as observation requires a population of cells that starts S phase at the same time. To synchronize the cells, the first step involves treating a population of cultured cells with a specific drug that prevents them from entering the S phase but allows them to progress through the rest of the cell cycle until they reach the end of the G1 phase. In the second step, the drug is removed from the culture medium, which allows the cells to resume their cell cycle. As a result, all the cells in the culture enter S phase simultaneously. The cells are synchronized for at least one round of the cell cycle. Now let's explore the type of experiment that can be conducted on these synchronized cells. The first step is to perform a pulse-chase BrdU incorporation experiment. BrdU, or bromodioxyuridine, is a structural analog of thymine that can be incorporated during DNA synthesis. During the pulse phase of the experiment, the AT-rich regions of the DNA that are actively synthesizing will incorporate the BrdU. Thus, the AT-rich sites of initiation of replication will be labeled, presumably at the AT-rich 13 mers. After the short pulse, a chase phase begins. At the start of the chase, the culture medium is completely removed and replaced with a medium that contains a very high concentration of deoxythymidine triphosphate, or DTTP. This means that any DNA that is synthesized during the chase will not be labeled with BrdU. The chase continues until the cells are fixed and processed for microscopy. Cell fixation involves treating the cells with a fixative agent that kills the cells while preserving them in the state that they were in at the end of the chase. After fixation, the cells are permeabilized, and the fixed, permeable cells are incubated with an antibody that specifically recognizes BrdU, which is referred to as anti-BrdU. This permeabilization allows the antibody to penetrate the cells and enter the nucleus. The anti-BrdU is conjugated to a fluorescent molecule, which allows it to be visualized through fluorescence microscopy. Sites that contain a high concentration of BrdU will become visible as fluorescent green spots. This visualization method can be used to detect replication sites that are active at specific times during S phase. This approach relies on the timing of the pulse, as a given origin of replication is only activated once during the S phase. What results would be expected when the BrdU pulse is performed at different time points during the S phase? If potential replication sites are not all activated simultaneously during the S phase, then a single pulse conducted at time points P1, P2, or P3 will yield different staining patterns. In these experiments, a BrdU pulse in S phase showed active replication sites after a 10-hour chase. This confirmed that not only can the sites be visualized, but software can also be employed to calculate the number of active replication sites or replication signals that were active during a specific time frame in an experiment. So, what did the patterns of replication sites reveal in this experiment? Experimental data have demonstrated that there are 3 types of replication sites that are activated at different times during the S phase. Each type of replication site corresponds to the replication of distinct regions of chromatin. Type 1 replication sites are activated during the early to mid-S phase and are associated with the replication of extranucleolar and euchromatic regions. The extranucleolar area is the region outside the nucleolus, which is a nuclear structure where ribosomal RNA is transcribed. Type 1 replication sites exclude replication within the nucleolar chromatin and focus instead on euchromatin, which is the less condensed portion of chromatin that includes most transcribed gene regions. Type 2 replication sites become active during mid to late S phase and are responsible for replicating DNA regions surrounding the nucleolus. These sites are associated with perinuclear heterochromatin, which is located at the periphery of the nucleus. Heterochromatin is the condensed form of chromatin that is transcriptionally inactive. Perinuclear means situated around the nucleus, while perinucleolar means situated around the nucleolus of the cell. Type 3 replication sites are activated late in S phase and are responsible for the replication of satellite DNA. Satellite DNA consists of long stretches of DNA that contain numerous short repeated sequences, which are usually non-coding and do not code for proteins. Now we can pose another question. What's the average time of activity for a single replication site? To measure this average activity time, a modified version of the pulse chase experiment can be employed using synchronized cells. This method utilizes two analogs of DTTP, BRDU and CLDU, chlorodeoxyuridine. These have the corresponding antibodies, anti-BRDU and anti-CLDU, that are labeled with distinct fluorescent markers. The experiment consists of 3 labeling steps. In the first step, a pulse of BRDU is administered at a specific time during S phase, starting with synchronized cells. The second step involves a chase period of variable length, typically less than 8 hours, which aligns with the duration of S phase. In the third step, following the initial chase, CLDU is introduced, and a subsequent chase is conducted until the cells are fixed and stained with the respective antibodies. This method lets us determine the average time that individual replication sites are active by analyzing the incorporation of both analogs during S phase. This representation illustrates 2 experiments that differ in the length of time between the 2 pulses, or the chase period between steps 1 and 3. In the first experiment, there are 3 replication sites, labeled RS1, RS2, and RS3. Each is active at different times during the S phase. RS1 and RS2 are both active during the BRDU pulse, while RS1 is inactive during the CLDU pulse. In contrast, RS2 and RS3 are active during the CLDU pulse. The expected results are as follows. RS1, having incorporated BRDU during the pulse, will show up as a green spot, since the anti-BRDU is labeled with a green fluorescent marker. RS3, which was active only during the CLDU pulse and not the BRDU pulse, will appear as a red spot because the anti-CLDU is conjugated with a red fluorescent molecule. On the other hand, RS2, which was active during both the BRDU and CLDU pulses, will be recognized by both antibodies and will exhibit a combination of green and red fluorescence, which appears as a yellow fluorescent spot under microscopy. In the second experiment, the timing of the BRDU and CLDU pulses during S phase are altered. Only RS1 is active during the BRDU pulse, while RS2 and RS3 are active only during the subsequent CLDU pulse. Due to the increased chase period between the BRDU and CLDU pulses, RS2 is no longer labeled by BRDU and incorporates only CLDU. As a result, RS1 appears as a green spot, BRDU only, while RS2 and RS3 appear as red spots, CLDU only, instead of yellow. This highlights how the increased chase ensures that RS2 is exclusively marked by CLDU. The average time of activity of a replication site is determined as the minimum length of the chase period between the BRDU and CLDU pulses that results in distinct green or red replication sites without yellow spots. Yellow spots represent an active replication site during the 2 pulses. Experimental evidence shows that on average, a replication site is active for approximately 45 minutes during S phase. We can do a calculation to explore the relationship between replication sites and origins of replication. We know that during the S phase of the cell cycle, the entire genome, which is approximately 10,000 megabases in size, is replicated over a span of about 8 hours, or 480 minutes. Experimental data show that each replication site is active for an average of 45 minutes, and that around 1,000 replication sites are active simultaneously during each 45-minute interval. This means that over the course of the S phase, there are 10 such intervals, which results in a total of 10,000 replication sites being activated. Each replication site is responsible for replicating approximately 1 megabase of DNA. Therefore, to replicate 1 megabase, it would theoretically take 300 minutes. However, this poses a contradiction, as a single replication site manages to replicate 1 megabase in only 45 minutes. To resolve this discrepancy, it is hypothesized that a replication site must contain multiple origins of replication. If we consider that each origin replicates 150 kilobase pairs in 45 minutes, a single replication site would need at least 6 origins of replication to achieve the observed rate. This means that, during S phase, a replication site consists of around 6 origins that work simultaneously to enable the replication of 1 megabase of DNA in 45 minutes. The next question we can pose is whether a replication site that is activated at time t during the S phase also replicates at the same time t in the next cell cycle? To address this, an experiment is conducted using a double pulse labeling approach, not within the same S phase, but across 2 consecutive cell cycles. The procedure begins with synchronized cells, where a BRDU pulse is administered 10 minutes after the start of the S phase. This pulse labels active replication sites, such as RS1, with BRDU. Following this, a chase period longer than 10 hours is performed to ensure that the cells progress beyond the end of the first S phase. The cells are then synchronized again to bring them to the same stage in the second cell cycle, ready to enter the next S phase. 10 minutes after the start of the second S phase, instead of a BRDU pulse, a CLDU pulse is applied, followed by another 10-hour chase period. After the second chase, the cells are fixed, permeabilized, and stained with antibodies specific to BRDU, labeled with a green fluorescent molecule, and CLDU, labeled with a red fluorescent molecule. If the hypothesis is correct and a replication site like RS1 is activated at the same time in both cell cycles, it would incorporate BRDU in the first cycle and CLDU in the second cycle. As a result, it would be detected by both antibodies and show up as a yellow fluorescent spot under microscopy. The experimental results showed that 100% of the replication sites appeared as yellow spots, which indicated that the replication sites were activated at the same time during both cell cycles. This confirms that replication timing is consistent across cell cycles and supports the conclusion that the timing of replication site activation is preserved from one S phase to the next. By visualizing replication sites, we have learned that these sites are activated in a coordinated manner throughout S phase and remain consistent across cell cycles. Each replication site comprises at least 6 origins of replication and serves as an illustration of the spatial organization of the genome within the nucleus. This coordination and structural organization play a crucial role in ensuring accurate and efficient DNA replication.